We have been talking about ECM, and we have talked a lot about ECM. I hope I haven't given you an impression that ECM is a static structure. It does not change, or it doesn't have the capacity to change. If that were the case, a lot of physiological functions that our tissues perform, they will be hindered. So ECM has to be degraded when required. For example, during embryogenesis, we have webs between our finger. We also have a tail. Both these tissues are degraded, and these tissues also contain ECM besides cells. So remodeling of ECM is a very important aspect of ECM physiology. So let's see how ECM is degraded and what requirements have to be met for that. Localized degradation of matrix components is required when cells migrate through basal lamina. I hope that's obvious. We have talked about this before too. When part of our tissue is damaged, and also, for example, if it is infected, our immune cells have to be able to access that area. Now, since our body is compartmentalized by this ECM, these cells will not be able to enter those domains which require their attention unless they have mechanisms that would allow them to degrade ECM specifically and enter those regions. White blood cells, for example, migrate across basal lamina of a blood vessel into tissue in response to an infection or an injury. Cancer cells, there are different types of cancers. There's a benign type and there's a malignant type. The difference between these two types is basically the malignant cancer can spread and go to different body parts, whereas benign cancers or benign tumors, the cells just grow indefinitely. Of course, the tumor is not removed, but these cells don't have the ability to spread and go into different regions. So cancer cells also have the ability, if the malignant cancer cells have the ability to degrade ECM and move to a different location, this process, of course, is known as metastasis. The seemingly static extracellular matrix of adult animals, even in adult animals, there is slow and continuous turnover, with matrix molecules being degraded and resynthesized. I told you the life of collagen in our bones, that this is an exception, that it is about the turnover rate for collagen molecules in our bone is about 10 years. Okay, whereas when we were talking about scurvies, we said the collagen in which holds our teeth in place, in the correct place in the socket, that has a lower turnover rate, and unless new collagen is produced, these teeth fall off, which happens, for example, in case of scurvies. ECM components are degraded by extracellular proteases that are secreted locally by the cells. Proteases belong to either of one of these two categories. One of them is matrix metalloproteases, and they depend upon the availability of metal ions, either it is a calcium or a zinc ion. And the second category is serine proteases, which have a highly reactive serine in their active site. Serine is an amino acid. We have talked about those. Both proteases cooperate to degrade ECM. Some metalloproteases are highly specific, cleaving particular proteins at specific sites to ensure the structural integrity of ECM is retained. But it still allows the cells to migrate, and it also these enzymes facilitate a small amount of proteolysis. Some metalloproteases are less specific, but they are anchored to plasma membrane and they get activated where they are needed. So, for example, the plasma membrane of white blood cells can have these specific enzymes that will allow them to cut through the ECM. Cells that migrate readily on type 1 collagen can no longer do so if the collagen is made resistant to proteolysis, for example, by mutating the sites on collagen molecule that are sensitive to cleavage by these enzymes. The proteolysis of matrix proteins can contribute to cell migration in 4 ways. It can simply clear a path through the matrix, so cells can traverse the matrix and go to a different location. It can expose cryptic sites. So if you cut a protein molecule, it can expose a sequence of amino acids that can that basically can promote cellular binding and allow these cells to now stick to that region, whereas they could not stick to that region previously. It can also promote cell detachment, so the cells can move onward, or it can release extracellular signaling proteins, stimulating the cells to migrate. The local activation: many proteases are secreted as inactive precursors that can be activated locally when needed. For example, plasminogen is an inactive protease precursor and is abundant in our blood. So if this enzyme was in an active form, it would do a lot of damage. And there are instances where we need a protease that has the ability to degrade ECM right at a very short notice, right there and right then. So the problem is solved by having plasminogen available in blood in enough quantity, but it is an inactive form. Plasminogen is cleaved locally by plasminogen activators to yield active serine protease plasmin, which helps break up blood clots. You may have heard of tissue type plasminogen activator, abbreviated TPA. It is given to patients who have just had a heart attack or a stroke. These two events, in many cases, result from formation of a clot. So when this enzyme is given, this activates the protein, the enzyme, which has the ability to degrade ECM, allowing the clot to dissolve away, restoring the blood supply to the patients. Confinement by cell surface receptors. Many cells have receptors on their surface that bind proteases, thereby confining the enzymes to the sites where it is needed. A second type of plasminogen activated, called urokinase type plasminogen activator, abbreviated appropriately UPA, is found bound on receptors of growing tips of axons that are leading at the leading edge of these axons, and also some migrating cells, where it may serve to clear a path for their migration. So basically, you can think of these proteases on surface of cells that need to migrate as little saws, which can cut through the ECM and clear the way so these cells can migrate. Receptor bound to urokinase type plasminogen activator also help cancer cells to metastasize. So let me show you an experiment. Here we have a cell which is secreting active protease UPA, and this UPA binds the cell receptors, and cells can grow and metastasize. If we take the cell and we transform the cell, start expressing a type of protease receptor, which is a mutant form of UPA that can bind the receptor but cannot cleave the proteins. And in this case, this protein, the mutant UPA, is produced in abundance as compared to the active protease. Now, both these cells are capable of producing active proteases. Here, the probability is majority of the protease that will bind the receptor is the mutant form, since it is produced in larger quantity. So this type of cell can metastasize, but this type of cell, although can form a tumor, can grow, but it cannot metastasize. So again, demonstrating that interaction of these UPA, these proteases, with the receptor is essential for their activation. Secretion of inhibitors. The action of proteases is confined to specific areas by various secreted protein inhibitors. For example, tissue inhibitor of metalloproteases and serine proteases inhibitors, which are known as serpins. The inhibitors of protease specific, these inhibitors bind tightly to activated enzymes and block their activity. Inhibitors may be secreted by cells that at the margin of areas of active protein degradation in order to protect uninvolved matrix. They may also protect the cellular surface proteins, which play a very important role. We have talked about them. For example, they are used for signaling. So this basically concludes our discussion on ECM, and we have already talked about cell adhesion. We are going to, in the next module, start talking about cytoskeleton.